All right, everybody. Good afternoon. I think it's about that time, 1:30. Thanks for joining us today for our Tech Talk. I'm here to talk about the radiation sterilization technology. More specifically, I want to dig into dose range, have an open, honest discussion from the perspective of your contractor radiator about what might be an ideal dose range and how that can really benefit you if you're in the need for irradiation services. Wrong direction. So my name is Mike Flanagan. I am a radiation technical advisor for the Sterigenics Expert Advisory Services team. I've had the privilege of working for Sterigenics for over 17 years. I've worked for our contract laboratory, our ethylene oxide facility, and our radiation plant, and kind of put that all together into something I enjoy, which is learning about customers' product and helping them through the validation process and supporting our radiator sites. My bio is up here. If you're a little bit interested more about me, basically just says I'm a big science nerd. All right, so I put together the following agenda for today. You can't really escape one of my presentations without first starting with the relevant industry standards. Then I want to get into it and start painting the picture. What's the status quo from the perspective of a contractor radiator? What kind of dose ranges they're seeing? And you know, there's no right or wrong to that, but we want to see how that feels and what challenges there are. Then we'll go into a little bit of a kind of a review, kind of seminar kind of feel. We'll talk about minimum dose establishment, review those different options that are available. We'll move into maximum dose establishment. There isn't a great deal of information out there in some of the guidance standards, so hopefully we can dig into that a little bit deeper. Lastly, we'll wrap up with delivery of dose, also known as dose mapping, how we're going to apply that minimum and maximum dose to your product, and why it's an important phase. It's a little bit, I would say, honestly hidden from the manufacturers because it's largely executed and reported by the radiator, but it's definitely something you should be aware of when you're using radiation sterilization. And then lastly, we'll open it up for open discussions. I'm a pretty casual guy, so feel free to stop me along the way and ask me questions. I'm really here to give you that perspective on radiation. All right, so my key takeaways for everybody is hopefully you're all here because you're either interested or you know that radiation, gamma, X-ray, and E-beam, they're reliable, they're established, they're proven method for reducing microorganisms on your product. That should be a slam dunk. And then I want to talk about the establishment, let you know what I feel an appropriate sterilization range, and then more importantly, you want to know what the benefits that are going to be for you if you need those services. All right, we're going to talk about those relevant industry standards. We'll rip this Band-Aid off real quick. So the ISO 11137 family of standards, part 1, 2, 3, and 4. These are the guidance documents for the radiation sterilization technology. Again, both gamma, electron beam, and X-ray. Part 1, real high-level guidance document, takes you the whole life cycle from product process definition to the validation steps into routine processing control. Part 2 gets a little bit more meaty, a little bit more detailed, talks about establishing that minimum sterilization dose. Part 3 is the guidance on dosimetric aspects. Really what this one is all about is IQ, OQ, PQ for contractor radiators. Talks a lot about dosimetry, more useful to me than perhaps you as the manufacturer. And then we have part 4 coming. This is a document that's in a draft form currently, and it's set to be published later on this year. Again, another document I'm more excited about than you, but it talks a lot about it, calculating process uncertainty, how to use that to establish your routine monitoring and confidence intervals and statistical process control. So that's coming soon. All 4 of these documents, really good to have in your library if you don't have them already. So I'm gonna do one tangent in this presentation, one timeout, and here it is. I like to put presentations in perspective. I don't want you to just come in and say, where am I? What are we talking about? So I kind of created this sterilization life cycle. So your product life cycle is much larger than this. You've got design, development, testing, a whole lot of things happening. And then you've got this real tiny sterilization piece. So today we're going to be talking about maximum acceptable dose, minimum sterilization dose, and those kind of fit in the middle. You really want to think holistically about your product and your process. That's the way the FDA looks at everything. You want to understand how your raw materials affect your bioburden, your minimum sterilization dose, how it affects your maximum sterilization dose. So keep these things in mind. It's never too early to think about sterilization. Right in that design and development phase. So hopefully that gives you a little idea where we're at and where we're thinking and how to make sure you're thinking holistically about everything. All right, so let's talk about the status quo. Again, disclaimer, this is a little bit of my perspective. It's a little bit of a biased presentation, but that's probably a good thing. Give you an idea of how your radiator looks at everything. All right, so I need a little help with this one. So can anybody give me an idea of maybe a common sterilization dose or maybe a dose you're thinking about applying to your product? Maybe something you've already validated? Pardon? There you go. I did not pay him to say that, but there you go. 25 to 40. It's a very, very common dose range in the industry. I'd have to say how we got there was when the VDMAX methodology was published, there was a 15 kilogray or a 25 kilogray option. And 25 kilogray is a lot more forgiving. It's a lot easier to qualify for. So a lot of people start with 25. Where the 40 kilogray really comes in, it probably came from your contractor radiator themselves. They say they need at least this 15 kilogray window to dose your product. And that's really where we are for the vast majority of the products. But, the industry is getting a lot more complex. We're doing pharmaceuticals, liquids and powders. We're doing combination products. We've grown as an industry much beyond a simple polymer type product. So the reality is your radiator is looking at something like this. At any given day, in any given week, there's various VDMAX methods. There's method ones where product specific doses. You've got non-validated processes. Just a host of dose ranges that come into the radiator that they kind of have to sort. And they have to get those through the radiator efficiently to get your product back to you. So that's really the challenge. A little bit of a snapshot. I did an approximation of what type of validation methods have been used and what contract manufacturers approach Sterigenics with. It looks to be about a 60% for the VDMAX method. A little bit less, about 20% for method one. Those are probably more legacy type products for when those methods were more predominant. And then method two, it's pretty rare. It's mostly used for maybe like pharmaceuticals or anybody wants to get a really low dose range. That's maybe 5%. And then the other bucket is non-validated foods, general industrial products. So that's an interesting chart to look at. There's still a lot being presented to the radiator that they need to get through efficiently. So before I came here, I approached one of our radiator schedulers at one of our flagship sites, what I would call a main cell continuous flow radiator. I said, in any given week, tell me how painful your schedule is. And unfortunately, I couldn't fit it all on this slide, but in a very just generic sense, the week doesn't really start on a Sunday. The radiator is running all the time. So we'll just pretend we're restarting our radiator. This particular radiator starts pretty high. They start with their heavy density product. The density might change along the way, but you can see their doses are elevating a little bit. And they're using transitions to get from high density to low density to different types of dose ranges. So you can see each of these dose ranges, correlates to a cycle time. So it's going to phase. It looks like it's going to phase down, phase up, all throughout the week. We're going to do this big wave. That's pretty complicated. It takes a very good scheduler to accomplish that, to make sure you're using right transition types of product, to make sure we're meeting everyone's specification. I think in the perfect world, we'd stay the same cycle time all week, but that's not reality. So really what I'm trying to get across is it's quite a challenge in a given week to get through all these different types of dose ranges. So everybody kind of good with the status quo, what we're presenting at Contractor Radiators. All right, so let's just do a little bit of a recap on minimum dose establishment. There's quite a few options beyond 25 to 40 kilogram. We don't necessarily need to zero in on that because that's what everybody's doing. So a little bit of Jeopardy here. We just had that Jeopardy Champions episode. So the key contributor to the establishment of minimum sterilization dose might be. Anybody help me out? What's up? There we go. See, I did not pay him, but I might after this. So what is bioburden? Bioburden is the key to everything. This is the number of bacteria living on the surface that has not been sterilized. I'm good for one typo of presentation. Here it is. It's actually the number of bacteria and yeast and molds. You're talking about prokaryotic organisms when we're talking bacteria, gram-positive cocci, gram-positive rods, gram-negative rods. So you're talking about people bugs, soil bugs, liquid water bugs, and then your yeast and molds. So when you're talking about bioburden, lower is always better. So what are we really going to do to combat that, to give us the most flexibility when we get into minimum sterilization dose, is we're going to have good manufacturing control. I think that's really a slam dunk. So if you've got good control in your raw materials, your manufacturing, all your water systems, all your air handling systems, you're going to see a lot of different flexibility when we get to the minimum dose establishment methods. My little soapbox here, definitely characterize your bioburden. It's important to know the quantity for sure for the minimum dose validations, but you want to know what type of organisms there are. It costs a little bit of extra money to go that extra step, but it's definitely handy for problem solving and really just reducing your bioburden overall and see where it's coming from. All right, so we'll go through I think the most popular and simple minimum sterilization establishment method, which is method VD Max. This validates that the bioburden of your product is less than the resistance of a microbial population of maximum resistance. So kind of wordy, but it's, your bioburden is less resistant than the maximum resistance kill curves. So you want to reference the ISO 11137 Part 2 document for this, and also you want to supplement that with the AMI ISO TS 13004. So I circled the 25 kilogray. How to qualify that is you want your average bioburden to be 1,000 or less. That's, like I said, that's very forgiving. I think anybody with a reasonable manufacturing process can qualify that. 15 kilogray was the other popular option, but now that this TS 13004 is there's a lot of alternate options there for you. So you can think beyond 25 kilogray. That's really my point. We don't have to focus in on what everybody's always been doing. It's amazing if you qualify for a 22.5 minimum sterilization dose, what that can do for you. So I think as we go through this presentation, think about that irradiator schedule. Think about how many times your product in your dose range fits in that weekly schedule. I think your goal should try to fit into that schedule as many places as possible. If you looked at your dose range and your density only runs on Sunday and you deliver on Monday, that means your product's going to sit there for a whole 5 days before your contract irradiator can cycle back around to there. So that's kind of the thought process I want you to be thinking as we go through this. Method 1. It's the second method, so you're proving that your resistance is less than or equal to a resistance of a microbial population having the standard distribution of resistances. Again, it's an older method. It isn't used as much. We're not going to get into the details of it. It takes more samples, more funds, more time. So it does have some benefits, though. It's going to give you a product-specific dose. And there's alternate sterility assurance levels that you can validate if you're not necessarily looking for the full 10 to the negative 6 sterility claim. Kind of circled the top there. You can see those alternate sterility assurance levels. And if you've got an average bioburden of 1, you can get down to 14.2 kilogray. That's a little bit less than a VD Max 15. So again, reference 11137 Part 2, Section 7. Last, probably not a heavily used method, it's method 2. This is a pure resistance type of validation. You're proving that you're using incremental dosing on your product to make sure that your radiation resistance is there. So again, reference ISO 11137 Part 2, Section 8. I'm not going to spend a whole lot of time on this. It's the real benefit of this one is you can get down to about the 8 to 8.5 kilogray range if you have some really sensitive products. So we covered there kind of the low end of the spectrum. We're going to try and use my football analogy. We're going to build our goalposts. So that'll be our left-hand goalpost. We know what our minimum sterilization dose is. Now let's talk about maximum sterilization dose. Before I can really do that, I have to kind of explain the concept of dose uniformity. So when you irradiate a product, you can't irradiate to a pinpoint dose. You can't give 26 kilogray, you can't give 30 kilogray. There's always going to be a range of dose around the product when you expose it. So Dose uniformity, DUR, is the ratio of maximum to minimum dose in the irradiation container that goes through the site. I just have a real generic example set of data here. You can see, I don't think I've got a laser pointer, but you can see there's 13 positions in this dose map. The nomenclature isn't important, but we do dose maps in triplicate and average the data. Kind of plotted it out for you here. It looks like I've got like a method 2B kind of dose range here. We've got about an 8.5 to 15 kilogray. Those are the top two lines. Excuse me. And you can kind of plot the data for this example product along the way to see how it kind of fits between those two goalposts. So you can think of this, and it'll show up later in the presentation, but you can kind of use that cooking analogy, kind of cooking the turkey. There's always going to be cold spots, there's always going to be hot spots, and the same kind of concept is there for irradiation. So for this example, I've got an overall max of 14.1 divided by the overall minimum of 10.8, which gives me a dose uniformity of 1.31. All right, so maximum dose, I think it's an often overlooked validation piece in the industry. They kind of sneak it in there, and there's not a whole lot of information, but compliance to the standard requires you to go through this experimental program so you can show that when treated with the maximum dose, the product is functional throughout its defined lifetime. That's probably also termed your shelf life. And it's also suggested that you do this on test product that's been irradiated to doses equal to or greater than your highest dose anticipated during sterilization process. So if you're thinking 25 to 40 kilogray, you want to be doing this max dose testing, probably 45, 50, 55 kilogray, proving that worst case scenario. Because we just talked about dose uniformity, so when you apply a dose, there's always going to be that range. So if you target 40 kilogray, you're probably going to get a dose range to these sample products between 35 and 45. You want to be thinking above and beyond your maximum sterilization dose. All right, so I had a bullet there that what's your highest maximum dose expected during the sterilization process? And a lot of that has to do with the capability of your irradiator. So this is an example. It's kind of, that's the way I look at dose uniformity. So what's the capability of your irradiator? And if you think in terms of a gamma total irradiator, they can probably get DURs probably about 1.5 or greater. When you do a dose map, typically dose uniformities are 1.1 to 1.3, but your irradiator needs a little bit of buffer. They need there's uncertainty in the statistical process. When you flip to a gamma pallet irradiator, you're talking about a bigger cube of product, much more penetration width, so the DUR is going to widen a little bit. It's probably on the scale of 2.0 or greater. Now, electron beam, we're going to spend a little bit of time on that today, but this is highly product dependent. You can get some really excellent dose uniformities on E-beam with certain products, but in general, if you're putting something more complicated, high density polymers, if you put any liquids or powders, you're probably going to see pretty extreme dose uniformities on that, probably 2.0 or greater. So there isn't a great example I can give you that. I've seen anything from a 1.1 to a 3 DUR for your electron beam. All comes down to presentation and what you're dealing with. X-ray, very similar to gamma technology. You're going to get DURs in the 1.5 range. So where I'm going with this is if you've got an example product, you think your minimum dose is going to be 25 kilogray, and you're gonna approach a gamma total irradiator, they're probably going to ask for 40 kilogray because that's where that 40 comes from. 25 times a DUR of 1.6, you're going to get 40. The gamma pallet irradiator, they're probably going to ask for 55. Same with an E-beam, and an X-ray is going to be pretty similar to the gamma technology. So again, I'm just trying to hit this point home. If you've got that 25 to 40 kilogram dose range, you definitely want to be doing your max dose testing, probably much beyond that, 50 to 55 kilogram. So you've gone well beyond what you're going to see in the routine processing environment. All right, I've been talking a while. No one's asked any questions. How we doing? Okay. We're going to get into a little bit of a sciencey stuff. I'm going to talk about the delivery of dose. If you're not too familiar with a gamma irradiator or electron beam irradiator, I'll give you an example of a gamma here. Every gamma irradiator has a concrete bunker filled with a source, which is radioactive cobalt-60. Every time an atom undergoes disintegration, it's giving off 2 photons at 2 different energies. And You can see in the center of our diagram here is our source rack, and we're going to pass totes of product, carriers or pallets of product, and we're going to zigzag it past this this source rack to give you the best even distribution of dose as possible. Again, that cooking the turkey analogy. Gamma is electromagnetic radiation, photons of pure energy, great penetration. So this is isotopic radiation coming out of the source rack. I'll just do one of my nerdy things. I like context. So there are millions of curies of radioactive cobalt in these bunkers. So a single curie has 3.7 times 10 to the 10 atoms undergoing disintegration every second. So you can multiply that by that 2, because we know there's 2 photons coming out. So 7.4 times 10 to the 10, and then multiply that by the millions of curies in the bunker. I think it's kind of an astronomical amount of radiations coming out. So where I'm going and where I'm building here is we're going to talk about dose mapping. So it's important to understand how we're applying this dose isotropically. So when we present product, we're going to present it in such a manner so we can get that nice max to min ratio on the product. Quick overview of E-beam and X-ray. Here's an example site. You've got your accelerator here kind of on the top level. I won't go through the physics of that, but we're going to generate electrons. We're going to collect them. We're going to give them a charge. We're going to give them speed, a whole lot of energy. We're going to bend them down and paint them onto the product. So it's highly directional. It's no longer isotropic technology. We're going to be pasting on the product. We're going to be passing product number probably on the scale of shipper boxes or small totes. The difference between electron beam and X-ray is that we put an electron converter in between the ray and the product. Basically, it's something of a high molecular weight, like a tantalum. So what that does is it slows down the electrons, and the energy has to go somewhere. So it gives it off in the form of an X-ray. So we get a lot deeper that in other seminars, but there's a kind of your 10-second explanation for an E-beam and an X-ray. Again, the main difference being highly directional. All right, so now we get into my favorite part of the technology, which is performance qualification dose mapping. So coming into this process, we've characterized our irradiator, we've done our IQ, we've done our OQ. We understand from a high level what its capability is. We've walked through how to establish your minimum sterilization dose. We've walked through the maximum sterilization dose, and we've built those goalposts. So now what we're going to try and kick through those goalposts is your product. That's our football. So we want to know, and we want to be upfront, that partnership. Talk to your irradiator. You want to understand what the overshipper dimensions are, what the overshipper weight is. We'll talk even deeper about what's the product heterogeneity. Is it, common polymers? Is it quasi-homogeneous, similar density, uniformly distributed? Or do we have non-homogeneous products? Do we have metal implants? Do we got liquids? Do we got powders? And if there's any temperature sensitivity, if we've got any dry ice requirements, all those kind of, build a recipe for your PQ. How we present the product to the irradiator to get that narrowest min to max ratio on the product. So I say, even though this process is a little bit invisible to a manufacturer, it's very critical. It's very important that you kind of pick their brain, how they're going about qualifying your product, because it's going to define your product configuration. It's going to define what's your expected minimum and maximum dose zones. It's going to talk about routine processing, how we establish our dosimeter placements. You're really going to understand what your dose uniformity is and how it fits in those goalposts. And then you're going to have a projected cycle time for future routine processing. So I'm kind of wrapping up here. I would say when you're coming into this process of validating for gamma, X-ray, E-beam, first and foremost, you want to think about that bioburden. You want to think about the quantity. You want to think about the morphology, what kind of resistance they might have. That's going to help you understand where you're going to select your minimum dose. Then think about your product and packaging design. Real high level, real important focus on materials. You want to know if there's any radiation effects, if you got anything that's sensitive, if you have any sensitivity to temperature. I would direct you back to an excellent reference, which is AMI TIR 17. That's where you're going to get a lot of your materials information, a lot more details that we can't dig into today. There's also some Sterigenics literature that you can pull off the website about materials consideration. And there's a host of webinars myself and my team have done that you can listen. We've done about an hour-long webinar on maximum dose. I've done one on a performance qualification. So we had real high surface today, giving you an idea, because we're just really talking about dose ranges. But if you want to dig into each of those topics, those resources are there for you. All right, so the key takeaways I want to leave you with today. So what I want to sell is there's significant benefits to thinking about your minimum dose options and your maximum dose establishment, getting the most reasonable dose range possible. How you're going to do that, you're going to have good manufacturing control. You're going to establish the lowest possible minimum dose that's reasonable for you. And then you're going to establish a maximum dose that's thinking above and beyond what you're going to see in routine production. That's going to help you with your product packaging design and functionality through the lifetime. And the benefits, here's what we're all here for. It's really going to help your turn time. Think back to that irradiator schedule. Think about how you can fit in there more times a week. That's going to reduce your overall sterilization costs. So, we're talking about time and money. That's good things, right? From a quality perspective, you can mitigate processing deficiencies. If there's still sufficient room in your dose range after a product's been dosed, maybe a little bit incorrectly, you can augment that dose with plenty of room where you're not exceeding any maximum dose. So there's a good quality perspective there. There's a potential for 2x processing. And mainly, I think what we're here for, as an industry, is we want consistent, reliable, robust process so we can deliver, functional and awesome medical devices for your end users. So again, I'm going to bring back my irradiator schedule here. So here's a perfect world. You have a little bit of a wider dose range. You might fit in 4 or 5 times a week, whereas maybe right now you're only able to process 1 day a week if you've got a really challenging dose range or a really challenging density. So that's all I got. I think there's probably still plenty of time. If you've got any questions for me, even if it's not topic related, I live and breathe the radiation technology every day. So hit me up now or maybe after if you don't want to poke me while I'm standing here. Okay. Well, I appreciate you all coming. Hopefully you took away some fun facts that maybe you'll get an advantage over the other guy. Think about that irradiator schedule. Think about how you can get in there more than 1 time a week. All right? You have a question? No, that's where I'd say you need to partner with your irradiator. So the question was, would every irradiator in the system follow that schedule? I would say no. Whoever your irradiator is, they're part of your manufacturing process. So partner with them. Understand what dose ranges they run, what types of products they run, and their densities, and they'll give you an honest opinion of how many times a week that'll fit in. There's, like I said, there's tote irradiator, there's carriers, there's batch irradiators, there's pallet irradiators, electron, E-beam. You really just need to give them a call, or I like to hear myself talk, so give me a call. I'll tell you all about the facility, what they got going on, and how your product might fit there, and how successful, how efficient it might be, and that all leads to turn time and pricing. Yes, so the question was, how does the design of the conveyor, the tote system affect the application of dose? Again, very irradiator specific. So they'll have a good idea when they do their qualifications. They fill up their totes, their carriers with dummy products. It'll be a foam, it'll be a cardboard, it'll be a ceiling tile, it'll be a wood. So they'll take all that data and thousands of dosimeter positions and readings, and they'll run statistical t-tests and p-tests on them and pick out the known highs and lows. A lot depends on how big the tote is. I could give you a general, high doses are always on the top of the tote, low doses are usually on the bottom, and they migrate towards the middle as it gets denser. But If you've got a big giant tote like a JS 10,000, so a lot of times the min doses are on the top. So again, it's that communication. Call the quality managers. We got one here. They'll give you how your product's going to get configured, where they expect the minimum and maximum doses is, and how it fits into their product mix. All right. Well, I'll probably loiter around for a little while. Come grab me if you have any questions. And thanks for joining me. Enjoy the show.